Welcome children. Now in this video, the latter half of protein synthesis, that's translation, you'll get a clear picture with video clippings. So you'll have a visual effect and this will make your studies more lively. Okay, so let's get started. The second stage of protein synthesis, translation. It involves 3 steps: initiation, elongation and termination, just like transcription. Now, initiation is a process where many things occur simultaneously. At this time, we have the mRNA with all its bases, and remember the bases are read as codons, 3 consecutive bases, and the first codon is AUG and the last is one among the stop codons UAA or UGA or UAG. And this is the region that is going to be translated. Now you can see in the cytoplasm this mRNA is present, having the start codon and any one of the stop codon. At the same time you can see a number of tRNAs. Now I am indicating the tRNAs like this. Then a number of amino acids also are present. I am going to represent in the form of circles. And don't forget the amino acid that starts the protein synthesis, met. Somewhere here I am putting met. Then introduce the other amino acid, proline, lysine, arginine, leucine, serine, phenylalanine, valine, glutamine, etc. etc. So 20 amino acids, a number of tRNAs. Then we have a mRNA, remember, with the start codon and the stop codon. Okay. Now what happens in initiation is, first of all, the ribosomal unit, small and large, comes and binds at the start codon. First the small ribosomal comes and binds, and then the large ribosomal comes and binds. Simultaneously, the tRNA, which tRNA? The initiator tRNA having the anticodon UAC. UAC is complementary to AUG. This tRNA is called the initiator tRNA. This initiator tRNA binds with the amino acid methionine. So this will bind with the amino acid methionine. See, with methionine. And this complex comes and binds here. Okay. And then there is the codon anticodon base pairing. Okay. This forms the initiation complex. This is how initiation starts in translation. So it's a very simultaneous process. So when you explain, you must say at the start codon what first comes? The small ribosomal unit. Then covering it comes the large ribosomal unit. Along with that, at the same time, what happens is the initiator tRNA having the anticodon UAC charged with the amino acid methionine comes and binds. All together happens, and this forms the initiation complex or the translational complex. This is how translation is initiated. And the remaining steps we will watch when we are watching the video. I will give a running commentary and you can visualize the steps as we proceed. After seeing the video, we can get back to the slide. So we have the mRNA in the cytoplasm with the start codon AUG and stop codon. Now the small ribosomal unit comes and takes its position at the start codon. Meanwhile, the tRNA charged with the amino acid methionine, this tRNA should be the initiator tRNA. So it has the anticodon UAC because the codon here is AUG. Now what happened? This UAC, this complementary base pairing between the codon and anticodon. This is the initiator tRNA. Now you see the large ribosomal unit binds. This forms the initiation complex or the translation complex. Now you can see 3 sites on the ribosomal unit. They are E, P and A. E stands for exit of the tRNA, P stands for peptide bond formation, A stands for the acceptor site. Now you see the new tRNA comes with a new amino acid charged with an amino acid. Now this new tRNA has anticodon which is complementary to the codon, and this tRNA binds with an amino acid And this binding of the tRNA with the amino acid is called charging of the tRNA or aminoacylation. It comes at site A. Meanwhile, what happens is there is a peptide bond formed at the P site between the two adjacent amino acids. Now the ribosome shifts from codon to codon. Now at E site the first tRNA exits. Now at A site a new tRNA comes in. Peptide bond is formed. Ribosome shifts. tRNA exits. New tRNA enters the A site. Peptide bond is formed. This continues until the stop codon is reached. When the stop codon is reached, a releasing factor comes and binds at the stop codon, releasing the peptide chain, the ribosomal units and the tRNA. And finally, you see the polypeptide chain binds into a 3D structure to form our proteins. So from the video, you got an idea of how the initiation complex is formed. I told you it's a simultaneous process where the small ribosomal unit, the large ribosomal unit along with the initiator tRNA charged with methionine forms the initiation complex. Then you have to remember the two sites E and A. E is the site where the first tRNA exits and A is the site where the new tRNA is accepted. Meanwhile, the tRNA does its role. What is the role of the tRNA? Reads the codon, and after reading it binds to the appropriate amino acid. The binding of the tRNA with the amino acid is known as charging of the tRNA or aminoacylation. It is an energy expensive process. Now this tRNA comes at site A. When it comes in position at site A, the two amino acids, adjacent amino acids, there is a peptide bond formed. After the peptide bond is formed, what happens? The ribosome shifts by one codon in advance or shift codon to codon along the mRNA. That time the first tRNA exits at site E. Again this process continues. New tRNA is accepted at site A. At site E, old tRNA exits. Meanwhile, peptide bond is formed between adjacent amino acids. This continues until the stop codon is reached. When the stop codon is reached, a releasing factor binds at the stop codon and it terminates the process of translation, and the polypeptide chain is released, the ribosomal units large and small are released and dissociates, as well as the tRNA is also released. This is how protein synthesis takes place. It is coming under the headings initiation, elongation and termination. Thank you.